Hello dear students. Today we will study about a very interesting topic, and that topic is DNA fingerprinting. Have you heard about this topic? Yes, just like our fingerprints which are present on our fingers, which are unique to us, they are our identity. Likewise, we have certain pattern on our DNA which are unique to us. Though you will be surprised to know that 99.9% DNA in all humans is same exactly. There is difference only in 0.1% of DNA. And based on the difference in the pattern of these 0.1% of DNA, we can make our DNA fingerprints. And the DNA fingerprints of only monozygotic twins, that is identical twins, is same. Otherwise it is different in all other organisms. We will study what are the different criteria, what are the different procedures, what are the various significance related to DNA fingerprinting. Let us study it. This concept or the technique of DNA fingerprinting is also called as DNA profiling. Only because we have a different pattern, we call them as fingerprints. Otherwise, these are not fingerprints as such. They are the sequences of nucleotides which are present. This technique was developed by Sir Alex Jeffrey very recently in 1985. So, these DNA profiling requires lot of processing, lot of technique to study and analyze the pattern. So, we will be studying the details of the DNA profiling. Before coming to that, we must know about this concept, that is DNA polymorphism. The complete DNA fingerprinting is based on DNA polymorphism. When I explained Human Genome Project, I told you about SNPs. SNPs are certain stretches of DNA which have got the changes with respect to only one or few nucleotides. Likewise, in our DNA, there are certain non-expressing DNA which do not code for any type of protein. So these non-expressing DNA, there are certain patterns, and these patterns are called repeats, or there are certain VNTR which are called variable number tandem repeats. These VNTR, that is variable number tandem repeats, are actually repetitive DNA which are specific for an individual. And we receive these VNTR from our parents. One VNTR for one particular repeat from mother and the similar from father. They have certain positions, they have certain locations on DNA. That's why DNA polymorphism is based on differences in these repetitive sequences at specific locus of the DNA. Okay, so this DNA polymorphism concept is very important. Again, I am repeating, DNA polymorphism is a type of variation shown by the repetitive DNA at certain locations. So, these concept of DNA polymorphism is based on variable number tandem repeats which are specific for a particular person. And these VNTRs act as the markers. We analyze only these VNTR when we do the complete profiling. And through the matching of these VNTRs of two different persons, we can come to a conclusion that the DNA which is extracted and profiled is of a particular organism or not. So, the DNA polymorphism is the backbone for the whole process of DNA fingerprinting. Okay. Now let us come to the significance of DNA fingerprinting. We can use DNA fingerprinting at various places. The main purpose is forensic science. By analyzing the crime scene, by identifying or collecting some type of human cells from that particular scene, we can later on profile that particular tissue, and then we can make the DNA profiles, and we can conclude and come to a conclusion that the DNA pattern is of a particular person, whether it matches to a particular suspect or not. Okay, so this DNA fingerprinting is used in forensic science. Likewise, DNA fingerprinting can be used for paternity and maternity test also, because an individual is a hybrid of his biological mother and father with respect to the DNA fingerprinting, with respect to the DNA pattern. Also, by studying DNA fingerprinting, we can study the evolutionary aspects of the different races or different types of individuals by studying the closeness and the variation in the DNA pattern. Okay, so this DNA fingerprinting is extensively used in forensic science, in paternity and maternity tests, also in studying the evolutionary aspects of various studies. Okay, now let's come to the procedure. It's a very extensive procedure, requires a lot of technical aspects of biotechnology. Let us first study the different steps which are used in studying the DNA profiling. Number one, this DNA fingerprinting starts with first step, which is isolation. Isolation of the tissue from the crime site or from a suspected person. Okay, so first it is isolated. Then after isolating and collecting that particular tissue or the blood, then it is subjected to centrifugation. Centrifugation means separation of DNA from the tissue. Here you can say this is the first step where tissue or blood samples, they are collected or isolated. Then it is subjected to centrifugation, second step, and the DNA is extracted. This DNA which is extracted from the crime scene or from the suspected person is very small in number. So it is first subjected for amplification with the help of PCR technique, that is polymerase chain reaction. This polymerase chain reaction we will study later on in detail. So this polymerase chain reaction is called gene amplification or gene cloning. Through this we get a large number of this extracted DNA. Then this extracted DNA is subjected to fragmentation, where that very large long DNA is fragmented into very small parts. That is done through a specific type of enzyme which are called as restriction enzymes. These restriction enzymes, they cut the DNA at specific points and specific fragments are produced. So the next step is the fragmentation, where these fragments of a long DNA are cut into small fragments, right? So this is fragmentation. Now, after fragmentation, those fragments are subjected to gel electrophoresis. Here, this is a technique where the fragments of DNA are separated from each other under the electric field. You can see this one. This is a setup of gel electrophoresis, where first of all a bed is prepared which is made up of agarose. So agarose gel bed is prepared. These are called as the wells, where the DNA which is fragmented is loaded, and this DNA is loaded under the electric field. DNA, because it is negatively charged, it will move towards the positive terminal. So you can see here it is cathode negative terminal, anode positive terminal. So from cathode towards the anode, these fragments will move under the electric field depending upon its size. The bigger molecule, the bigger molecule will remain close to the cathode, and the smaller molecules, smaller fragments, they will move towards the anode. So you can see the bands are produced depending upon the size of the fragments. So these bands cannot be seen, they are invisible. So they have to be made visible by another technique, in which first process is called Southern blotting. What happens once the bands are produced? These DNA band patterns are transferred to a nitrocellulose sheet or a nylon sheet, and this technique is called as, this blotting technique is called as Southern blotting. Okay, so you can see here this is blotted on this nitrocellulose or nylon sheet. After this is done, now whatever pattern has been blotted on the nylon sheet, to this is subjected DNA probes. These fragments are basically the VNTRs. So these VNTRs are also made into DNA probe. That is complementary. When we add here, actually what happens, after fragmentation, some alkali is added here. Because of that alkali, the double-stranded fragments are converted into single-stranded. So all these patterns which are here, they are single-stranded of DNA. Similarly, the similar DNA probes of VNTR are prepared, which are also single-stranded. When it is hybridized with this single-stranded, so wherever there is complementary base pairing, the pairs will be there. The hybrid DNA will be produced. So this hybrid DNA is subjected to X-ray film, because we are using radioactive DNA VNTR probes. Now when it is subjected to X-ray films, then the pattern will show the hybrids, and by studying this we can identify a specific type of dye is used, that is called ethidium bromide dye. This ethidium bromide dye shows the patterns, the colors of the bands, and when it is subjected to X-ray film, it can be seen. And then when we compare the bands which are produced with the suspected person, so wherever there is complete DNA matching, we can see that the person whose DNA is there, that will show a complete DNA hybridization. And through this, we can identify whether the suspect is actual criminal or not, or the paternity or maternity test is positive or negative. So this was all about DNA fingerprinting. This DNA fingerprinting is shown in your book also. Now we will switch over to your book, textbook, where this complete process of DNA fingerprinting is given. Let's see that. Let us now very quickly recapitulate the whole process which is given in your book. So this diagram shows the complete sequencing of DNA fingerprinting. You can see the patterns. There are patterns of two individuals, A and B, and which is compared with the pattern C. This pattern C has been identified from the crime scene, which will be called as VNTR probe, whereas A and B are the suspects. We will come back to it again. Let us first see the steps which are involved. These steps are: number 1, we have already studied isolation of DNA, followed by digestion of DNA by restriction endonucleases, then separation of DNA fragments by electrophoresis. After that, transferring or blotting of the separated DNA fragments to synthetic membranes such as nitrocellulose or nylon. After that, hybridization using VNTR probe. And finally, detection of hybridized DNA fragments by autoradiography. So we studied this whole process, and these VNTRs are nothing but they are mini satellites. So let us again come back to the same diagrams. Here you can see there are chromosomal pattern shown. So chromosome number 7 of two individual A and B. So in the chromosome, all organisms, all human beings, they inherit the chromosomes from their parents, paternal chromosome and maternal chromosome. So chromosome number 2 and chromosome number 16 are studied here. So after doing the gel electrophoresis, their patterns of their DNA are obtained, and these two patterns of A and B, when they are matched with C, which is actually the VNTR probe, we can see that the pattern of individual B matches completely with the person or with the crime scene pattern. So here we can identify that the criminal out of A and B is B. So this DNA fingerprinting is very commonly used these days in forensic science in identifying the criminal. Also, it is used for identifying the paternity and maternity test. Also, this is used in evolutionary studies to study the pattern of evolution in different races of human beings. So this DNA fingerprinting is a very important aspect. You must understand all the steps, all the terms which we have come across, and uh, understand and practice it very well for the exam, from exam point of view, because this is a very important question which is generally given for 3 marks or 5 marks. I hope you have understood it. Thank you.